Hello students, last class we studied about one biomolecule, that is carbohydrate, and here let us study about another biomolecule, that is protein. So proteins is derived from the word proteus. Proteus means vital. So this is very vital for growth and maintenance of life. In these proteins are very important. Okay, so these are the main structural material of animals, as cellulose is the main structural material of plants. Proteins are the main structural material of animals. Next, so chemically proteins are generally condensation polymer of alpha amino acids. These are the condensation polymer of alpha amino acids. Okay, so proteins are the condensation polymer of alpha amino acids. So now it is necessary to know what is amino acids. Okay, so after that we will go for proteins. So next come to amino acids. So amino acids, these are the organic compounds which contain functional group NH2 and COOH group. That is one amino group, one carboxylic group, that are known as your amino acids. And amino acids are different types like alpha amino acid, beta amino acids, gamma amino acid, depending upon the position of this amino group with respect to carboxylic group. Okay, so alpha amino acids, where the NH2 group present at alpha position. These are generally represented as NH2 CHRCOOH. Depending, so this is your carboxylic group, this is known as alpha carbon, and this is your NH2 group. So this is alpha amino acid, and this R group, that may be different types. Depending upon the variety of this R group, these amino acids are of different types or named into different types of amino acids. Okay, so look here, in this amino acid, this is the alpha carbon, and this is one side amino group, another side carboxylic acid group, another side this R group. So due to which these amino acids are also chiral in nature. This is the chiral carbon, that's why these are chiral nature, and these are optically active compounds. Okay, so all natural proteins are the condensation polymer of all natural proteins are the condensation polymers of L form of alpha amino acids. They are the condensation form of L form of alpha amino acids. Okay. Next come to how these amino acids are classified. So these amino acids are classified depending upon the structure, that is acidic amino acid, basic amino acid, and neutral amino acids. So acidic amino acids are there where two carboxylic acid is present. You know the group is NH2 CH COOH, or one carboxylic acid is present here. If there is another type of carboxylic acid present, another substituted carboxylic acid present here, that is known as your acidic carboxylic acids. Example, glutamic acid, aspartic acid, etc. So these are examples of some acidic amino acid. Similarly, basic amino acids, if this is the NH2 group and this group contains any other NH2 group, any other amino group, this is one amino group, and if this group again contain another free amino group or a substituted amino group, this is known as your basic amine. That means if it contains 2 amino group, this is known as basic. If it contains 2 carboxylic acid, this is acidic. And what are the basic amino acid? Example, histidine, lysine, etc. So these are examples of some basic amino acids. And neutral amino acid which contain only 1 amino group and 1 carboxylic acid, that is known as your neutral amino acid. Example, glycine, alanine, phenylalanine, etc. So these are some examples of neutral amino acids. Again, these amino acids are classified into 2 types depending on their synthesis. Depending on their synthesis, these are classified into 2 types. One is known as your essential amino acids and another non-essential amino acids. So far 26 amino acids are isolated from different proteins. Almost 20 amino acids are present in all proteins. Almost 20 amino acids are present in all proteins. Among the 20 amino acids, 10 amino acids our body cannot synthesize. It has to be provided from outside, from our diet. So the 10 amino acids that our body cannot synthesize, that our body can't synthesize. So the 10 amino acids that our body can synthesize are known as essential amino acids and are also known as your indispensable, also known as indispensable amino acids. Okay, so some examples of essential amino acids are valine, leucine, methionine, histidine. These are examples of some essential amino acids. Non-essential amino acid, the amino acid which our body can synthesize. So the amino acid which our body can synthesize is known as non-essential amino acid. So 10 amino acid that can synthesize by our body, that is known as your non-essential amino acids. Some examples are glycine, alanine, phenyl, glutamic acid, aspartic acid, etc. So these are the amino acids that are known as your non-essential amino acids. Next come to what are the properties of amino acids. So amino acid, due to presence of NH2 group and carboxylic group, amino group and carboxylic group, this behave as salt. So these are salt-like, solid, we can say crystalline. Okay. Next, these are soluble in water. Amino acids are soluble in water, insoluble in different organic solvents. And amino acids are generally amphoteric in nature. As these amino acids have NH2CHR COOH. As they have both amino group and carboxylic group, in this way this acts as acid as well as a base. Due to presence of COH group, this acts as a acid. Due to presence of NH2 group, this acts as a base. In this way, it is amphoteric in nature. An amino acid exists as zwitter ion structure. Zwitter ion structure. That means they have double ion structure. So at low pH, this amino acid acts as a cation. So it receives some H plus ion, that is acidic solution. In acidic solution, it receives some H plus ion. In this way, it acts as a cation. Low pH, it acts as a cation. And when electrolysis takes place, it moves towards cathode. If the pH value increases, okay, so if the pH value increases or at high pH or in basic solution, it acts as an anion and moves towards anode. So it acts as cation, move towards cathode, it acts as anion and move towards anode. In between this medium pH, at low pH or neither at high pH, in between this medium pH, it is different for different amino acids. In between medium pH, it exists both as cation and anion. This is known as your zwitter ion. And this this exists as in zwitter ion. So in this zwitter ion structure, if electricity is passed, it neither moves towards cathode or anode. And that pH is called isoelectric point. Isoelectric point. So amino acid possesses isoelectric point. What is isoelectric point? The pH at which the amino acids show no migration. The pH at which the amino acid show no migration, that is known as your isoelectric point. And at that isoelectric point, these amino acids are insoluble, or we can say less soluble. Here, these are less soluble. They neither move towards cathode and anode and exist as zwitter ion. Then they are less soluble and no migration, that is at isoelectric point. In this way, proteins can be isolated at isoelectric point. And due to deficiency of amino acid, diseases like kwashiorkor results. Okay. Next come to what happens when these amino acids combines. Okay, next come to what happens when amino acid combines. So you know NH2CHR COH plus, suppose another NH2CH COH R dash. So here OH of one acid combines with hydrogen of this amino group to form release of water molecule, condensation reaction. It forms NH2CHR CO NH CH R dash COOH. Okay. So due to when this chemically react or combines with itself, it forms another compound, and that is called peptide. So what are peptides? Peptides are also the condensation polymer of, we can say these are also condensation compounds or condensation polymers of alpha amino acids. So the sequence of combination is different, then the peptides are different. If two amino acids combine, then it can give if two amino acids combine in different way, then it can give 4 dipeptides. These peptides are classified into two types. One is oligopeptide, which contain few number of amino acid, that is 2 to 10. Okay, 2 to 10, that is known as your few number of peptides, and another known as your polypeptide. This is oligopeptide, and another known as your polypeptide. So polypeptide contain more than 10 amino acids up to 100, and oligopeptides generally contain a few number, that is 2 to 10 number of amino acids. These are known as dipeptide, tripeptide, tetrapeptide, pentapeptide, depending on the number of amino acids join or combine. The number of amino acids join or combine, to form these peptides. And when these combine, these form a functional group, that is CONH. This bond is called peptide bond. CONH. This bond is called peptide bond. So different amino acids are joined through peptide bond. Okay, so there is not much difference between this peptide and protein. Actually, peptides contain polypeptide and protein. Protein is also, we can say, a polypeptide. This is also a polymer of polypeptide. If the number of amino acid is more, then this is known as your protein, and the number of amino acids less, actually that is known as your peptide. Peptides on hydrolysis generally gives amino acids. Then, okay, so if the two peptides join, then it can give in different way. Then it can give 4 different peptides. For example, if A and B are two peptides, then it can form dipeptides with AA, BB, AB, and BA. If the sequence is different, then the peptide is different. For example, if we consider about glycine, this is glycine, and another alanine. So it forms the peptide. H is here and CH3 is here. So it forms glycyl alanine. So this is known as your glycyl alanine. So the name of the peptide is glycine alanine. If first you keep alanine and then glycine, this is known as your alanyl glycine. In this way, different types of peptides are there. Generally, different animal and plant toxins are our peptides. Next, come to another part of this, that is known as your protein. proteins. So, as previously I have told you that proteins are the condensation polymer of L form of alpha amino acids. Depending on the different types of amino acids taken, the proteins are different. This is not only one type of monomer unit is used here. So in a protein, different types of amino acids are joined. So in protein also, different amino acids are joined through peptide bond. It also contains peptide bond. Proteins also contain peptide bond. Different amino acids are joined through peptide bond. Okay. So these proteins are classified into two types. Depending on its nature, proteins are classified into two types. One, fibrous protein, and another globular protein. Depending on the structure, so depending on their nature, these are classified into two types, fibrous protein and globular protein. So fibrous proteins these are linear and fibrous nature. So the proteins which different proteins are combined together to form a linear shape or fibrous structure are known as your fibrous protein. Example, keratin, myosin, fibroin, collagen. So these are different fibrous protein. Keratin present in skin, hair, nail, myosin present in muscle, fibroin present in silk, collagen present in tendon. These are the examples of some fibrous protein, and these are insoluble in water. These are insoluble in water. Globular proteins. So the fibrous protein, if these are folded so many times, so that it can give a spheroid, different linear proteins. When these are folded so many times, so that it can give a spheroid shape. So that it can give a spheroid shape. So these are known as globular proteins. And globular proteins are generally soluble in water. Examples, albumin present in egg, albumin, hemoglobin present in blood, albumin, hemoglobin, maximum hormones, enzymes. These are examples of some globular proteins. Next come to another way. These proteins are again classified into two types depending upon the hydrolysis product. That is known as your simple protein and another conjugated protein. So the protein which on hydrolysis only gives alpha amino acids are known as simple proteins, and the proteins which gives alpha amino acids and some other vitamins like prosthetic groups. Prosthetic groups, these are known as your conjugated proteins. Okay. So these proteins, next come to the structure of proteins. Structure. So proteins structures are different types. One, primary structure. Covalent structure sorry, secondary structure, tertiary structure, and another quaternary structure. So the structures are classified into general structures are 4 types. Primary structure, it represents the covalent bonding of amino acid. So the condensation polymer, how is formed? It represents the sequence of amino acids. So it represents the covalent bonding of amino acids, but it represents the sequence of amino acids. Next, secondary structure. When these primary structures are formed, so different polypeptides, when this covalent forms, so suppose it forms some polypeptides. So this is the primary structure, only represent the sequence or the covalent bonding of protein molecules. Suppose there are different polypeptides, how these are arranged, this can be only known from this primary structure. And secondary structure. So after this formation of this primary structures, so if the protein is a long chain, then what happens? There is also along with this covalent bond, there is also another type of bonding, that is hydrogen bonding takes place. So due to this hydrogen bonding, if the hydrogen intramolecular hydrogen bonding takes place, if the hydrogen bonding takes place within the same molecule of protein, then what happens? The NH group of one amino acid, suppose there is a combination of different amino acid, NH group of one amino acid and CO group of the fourth amino acid. In a sequence, there are so many amino acids are there. So the NH group of one amino acid and the CO group of another amino acid. So there exists the hydrogen bond between this NH group and this CO group. As a result, what happens? Now this linear structure is disturbed and it forms a coil-like structure or helix structure. In this way, it forms a coil-like structure. So within if the intramolecular hydrogen bonding takes place within the molecule, then it has a coil-like structure. It forms, this is known as a secondary structure. And if different protein units are there, suppose there is one amino acid, polypeptide, and there is another polypeptide, this is one protein group, there is another protein, then what happens? The hydrogen bonding may take place between CO of 1 and NH of 1. Between NH of 1, CO of 1, NH of 1. So among them, this type of hydrogen bonding may take place. In this way, this quaternary structure, one has coil structure, helix structure, and another has beta flat or beta pleated structure. Beta flat or beta pleated structure. This is alpha helix structure. So these two are known as secondary proteins, and this is the primary structure of proteins. And another tertiary structure, this is the three-dimensional structure of this primary and secondary structure. This is the three-dimensional structure of proteins. So in this, the primary structure, the secondary structure is there. This is only 2D structure, and if the 3D structure is there, there exists another bonding like your, so this here covalent as well as hydrogen bond. And here there are different types of bonding like your sulfide bond. Due to existence of sulfide bond, hydrogen bond, and van der Waals force and dipole-dipole interaction, what happens? These molecules, protein molecules, they may folded in different way and give rise to different tertiary structure. So in this way, this is the tertiary structure of this protein. So this is the tertiary structure. And this tertiary structure, when this type of bonding is present, and again these proteins are folded so many times so that it gives a complex structure that is known as your quaternary structure. And this quaternary structure, so in this it folded so many times due to hydrogen bonding and due to sulfide linkage. Hydrogen bonding, sulfide linkage, van der Waals force, dipole-dipole interaction, etc. Due to this, the protein molecules folded so many times, and in this way it gives rise to a quaternary structure. So this is tertiary structure, this is quaternary structure, these two are secondary structure, this is primary structure. Next come to what is denaturation of proteins. So when the globular proteins, it exists in certain pH level. So when these when the globular proteins, the pH is changed, it makes acidic or it makes basic, or when it is heated by the application of heat, or when treated with some strong acids, or the pH is changes, then what happens? The biological and physiological activity of proteins lost. This is called denaturation of protein. So on application of heat, alcohol, acid, etc., the proteins lost their biological and physical activity, that is called denaturation of proteins. For example, you know egg albumin, that is the egg white. These are the globular proteins, and when these are heated, it becomes fibrous in nature. It becomes solid. So globular protein become fibrous. So this biological and physiological nature of protein is lost, that is called denaturation of proteins. Now, what is the importance of proteins? So proteins have so many functions. It is the structural material of animals. It helps for it acts as a transporting agent. So it acts as enzyme. Proteins are also used as different enzymes, and proteins are helpful for metabolic regulation. For example, insulin controls the sugar level, and thyroalbumin that controls the thyroid. Similarly, it acts as also, the proteins are also generate antibodies. These proteins also generate antibodies which prevent us for attack of different diseases. So it acts as antibodies. It is helpful in metabolic regulator. It acts as enzyme. Proteins are also helpful in transporting agent, and it is the structural material of animals. Next come to another part, another type of, we can say, protein, that is enzyme. So what are enzymes? Enzymes are actually, we can say, these are chemically all globular proteins. Enzymes are some globular proteins. These acts as catalyst in different biological process. That's why this is known as your biocatalyst. So enzymes are biocatalyst. It acts as a catalyst in different biological processes. Okay. So these are all globular. All enzymes are globular proteins. And the name of the enzyme generally depends on what type of substance it acts. For example, it acts if maltose is treated with enzyme maltase. The enzyme which acts on maltose to form glucose is known as maltase. Similarly, different enzymes are zymase, invertase, cellulase, urease, etc. So there are different types of enzymes, and these enzymes associated with some. So these enzymes are proteins, and these are associated with some non-protein groups. Enzymes associated with some non-protein groups that are called cofactors. Enzymes are associated with some non-protein groups that are called your cofactors, and these cofactors are two types. One is your inorganic cofactor, another organic cofactor. So inorganic cofactor, different metal ions like zinc, cobalt, manganese, copper, so etc. So these are some metal ions which are connected with this enzyme to increase the activity of enzyme. To increase the activity of enzyme, some other non-protein groups are joined with this enzyme that are known as your cofactors. So these are some inorganic cofactors. Similarly, organic cofactors. So there are different types. This is also two types. So the organic cofactor, which can be separated by dialysis, the smaller organic. We can see the smaller organic cofactors, the smaller organic compounds, or we can say smaller organic compounds. The smaller organic compounds which is associated with these enzymes that can be separated by dialysis, is known as your coenzyme. And the organic compounds which is associated with these enzymes to increase the activity, and generally that can strongly bonded. These coenzymes are weakly bonded, and the organic compounds which are strongly bonded with this enzyme and that can only separated by hydrolysis, that are known as your prosthetic groups. So when these enzymes are linked with this cofactor, that are known as your holoenzyme. And holoenzyme, after releasing of this cofactor, what is the enzyme form? That is known as your apoenzyme. So enzymes linked with cofactor form holoenzyme. After release of by dialysis or hydrolysis, it forms an apoenzyme. And enzymes generally it is very important for animals, for human being, and it acts as your biocatalyst. For example, if there is no digestive enzyme, then it requires so many years to digest food. In this way, enzymes helpful or it catalyze the different biological processes or different metabolic processes. So today up to this, next class we will discuss some other biomolecules.